Okay, I wanted to welcome everybody, both here in person and those watching on YouTube, to our Nano at Tech seminar. Before I introduce today's speaker, just a word. Our next seminar will be actually just next week, a week from today, instead of the usual 2 weeks, just to avoid conflict with another event. So our next speaker on April 5th will be Professor Carson Meredith from Georgia Tech's School of Chemical and Biomolecular Engineering. So it's a real pleasure to have with us today Professor Leslie Chan from the Department of Biomolecular Engineering. Leslie got her undergraduate degree in biomolecular engineering right here at Georgia Tech before going to the University of Washington to get her PhD in bioengineering and then doing a postdoc at MIT before returning here to good old Atlanta and Georgia Tech just last summer, where she is currently an assistant professor in the Department of Biomedical Engineering. She also just recently was awarded an NIH Pathway to Independence Award, which is a good way to get your career started off. And so I will hand it over to her, and we look forward to hearing what her research is going to be.
Can everyone hear me okay? Okay. Good afternoon. Thanks to David for the invitation to speak, and thank you to you all for your attendance. So as David mentioned, I'm an assistant professor in the BME department that's joint between Georgia Tech and Emory. And my lab works on nanomedicine approaches for diagnostic and drug delivery applications. So today I'll be discussing some of our work in breath-based diagnostics and how we've developed protease sensing nanoparticles that enable us to move forward with breath testing for clinical diagnostics. So human breath is a really interesting matrix comprised of thousands of volatile organic compounds, or VOCs for short. And when we exhale these VOCs, this is a clearance mechanism for the VOCs that are introduced into our bodies. And these VOCs come from several exogenous sources, such as our diet and the environment. And we also produce VOCs endogenously as metabolic products. And so there's this growing interest to try to identify which exhaled VOCs we can monitor in order to detect disease or to monitor disease non-invasively. But there's many limitations to current approaches to breath biomarker discovery that has really hindered the widespread use of breath testing for clinical diagnostics. And this has to do with both technical and statistical limitations with the current approaches. So generally, for breath biomarker discovery, we rely on clinical cohorts containing healthy and diseased patients. And through breath sampling and analysis using gas chromatography mass spectrometry, the goal with these studies is to quantify several hundred, if not thousands, of VOCs that exist in our breath, and to find which ones have differential levels between a disease state and a healthy state. And so with this type of approach, the technical limitation is that there's a trade-off between throughput and accuracy. And so we're talking about hundreds to thousands of VOCs through this GCMS discovery approach, in which case that really limits the amount of resolution, the accuracy you can have in resolving each of these VOCs. And then the statistical limitation is in the number of subjects in the clinical cohorts compared to the tested variables. So when the tested variables, which are your VOCs, are considerably larger than the number of variables, then you have a greater chance of what we call false voodoo correlations. And so for this reason, there are several VOC candidates that are identified but never really validated clinically because of these false correlations. And so because of these limitations, there are only a handful of clinical breath tests which rely on these naturally occurring volatiles for disease detection. And alternatively, we actually see a handful or a subset of these breath tests that rely on an alternative approach where the VOC is actually induced in patients through the introduction of an exogenous agent into the patient. And these exogenous agents are usually small molecule enzymatic substrates that are processed by disease-associated enzymes into volatile products that then eventually traffic to the lungs and are exhaled. So an example of this is the carbon-13 urea breath test, which tests for a Helicobacter pylori in the stomach. So this is a bacterial pathogen that can lead to chronic gastric inflammation and that can eventually lead to gastric cancer. In this breath test, patients ingest a small molecule substrate that's converted by the bacterial enzyme urease into a gaseous product. And because the substrate itself is labeled with a carbon-13 isotope, the resulting carbon dioxide is also isotope labeled, so that when it's exhaled, through this enrichment in the abundance of this isotope, we can determine if H. pylori is present in the stomach. And so essentially this breath readout is a functional readout for this enzyme activity produced by the pathogen itself. And as engineers, we find that this approach is more desirable than breath biomarker discovery approaches, because here we're leveraging known biological pathways to generate a known volatile that's eventually exhaled. And in several of these examples, the volatile that's being generated is a bio-orthogonal volatile, meaning that it does not naturally exist in the body. And so it's more easily discernible from all the hundreds to thousands of VOCs that are exhaled in breath. And so the current breath tests that leverage this approach is quite limited in scope because of the limited number of substrates that are available to be converted by disease processes into volatile products that are exhaled. And so with our experience in engineering enzyme-responsive materials, we really saw an opportunity here to engineer diverse arrays of enzyme-responsive materials to generate these breath readouts for disease. And in particular, we were interested in initially leveraging proteases, which are a subset of enzymes that are present in abundant quantities in the body. And these proteases act in concert to break down other proteins to maintain homeostasis during health. And this protease activity is generally regulated by endogenous inhibitors. So these inhibitors are also proteins, and they can bind the protease to prevent their activity. And when during disease, we oftentimes see an imbalance in proteases and their endogenous inhibitors that will lead to aberrant levels of proteases that can contribute to disease progression. And so for this reason, it's not enough to simply measure protease abundance levels because we're missing that inhibitor piece. And so for this reason, we're interested in measuring activity levels, which are a net result of this protease inhibitor interaction. And so with our prior experience engineering protease responsive nanoparticles, we were really interested in leveraging this class of enzymes for breath-based disease readouts.
Professor, is it more common for problems to be caused by excessive inhibitor or not enough inhibitor?
So based on the clinical diseases that we're focusing on, it's based on a deficiency in the inhibitor. But there may be examples where there's more inhibitor present than usual. Okay, so for us, we hypothesized that we could leverage protease activity to engineer these breath readouts by designing these nanoparticle sensors or nanosensors that carry a volatile reporter payload that's conditionally released in response to active proteases. And we predicted that we could engineer these nanosensors by strategically coupling volatile reporter compounds two protease cleavable peptide substrates in such a way that once these substrates are cleaved, they release the volatile reporter back into its original form so that it can recover its volatility to be detected as a gas. Furthermore, one of our goals was to optimize signal to noise ratio by increasing on-target delivery to diseased tissues. And we predicted we could do so by using nanocarriers to which we'd tether our peptide substrates. So for those in the nanomedicine field, you're familiar with the idea that nanocarriers can be used to modulate the distribution and the pharmacokinetics of certain cargo to after in vivo administration of your cargoes. So this is what we had initially planned to do in order to generate our breath readouts. Oh, sorry, that might be me. Okay. And so for our first test of feasibility, we targeted lung disease because obviously the lungs are directly responsible for generating breath. So this was a very obvious first place to start. And so we hypothesized that once we developed these nanosensors and delivered them into the lungs, we predicted that the substrates would be cleaved and that the reporters would be released into the alveolar space to be exhaled as a gas. And then we could subsequently quantify these reporter levels using portable VOC detectors or collect breath for downstream analysis using mass spectrometry. In our initial proof of concept, we decided to design a nanosensor for neutrophil elastase, which is a protease that's secreted abundantly during acute inflammation, such as that observed during lung infection or bacterial pneumonia. And so for this, we looked to the literature to identify a peptide substrate that was specifically cleaved by neutrophil elastase and that was also rapidly cleaved by the protease. And we were able to identify this peptide here. And using simple peptide chemistry, we were able to tether a volatile molecule to the C-terminus of this peptide substrate. And the C-terminus is where this substrate is cleaved by neutrophil elastase. So we predicted that upon cleavage, we'd have the release of our reporter molecule, which would then convert back into its volatile form to be detected as a gas. So we were able to confirm this using mass spectrometry. So in vitro, in a vial, we mixed our modified peptide substrate with the active neutrophil elastase, and then characterized the cleavage products that were in solution as well as those that existed in gas phase. And so using MALDI mass spec, we confirmed that, in fact, the substrate is cleaved where the reporter is tethered. And more importantly, once it's cleaved, the reporter rapidly transitions into gas phase and is detectable in the headspace of this reaction. And so these early studies were essential to demonstrating that we could, in fact, engineer a protease-triggered volatile release mechanism, which would then be important for proving our approach further downstream. So after validating our mechanism, we then formulated our modified peptide substrates for in vivo delivery. And we essentially tethered these peptides to a nanocarrier made out of polyethylene glycol, so an 8-arm PEG. And the idea here is that once it's tethered to the nanocarrier, these peptide substrates will be retained in the lungs rather than diffuse out of the lungs after direct intrapulmonary delivery. And so then we moved on to in vivo studies in our lung infection models, which have elevated neutrophil elastase activity. So we delivered these nanosensors and collected breath at various time points to see if we could, in fact, detect a signal from our nanosensors. So this graph here shows on the y-axis breath signal, which is the amount of released reporters exhaled in breath over time, where t equals 0 is the time at which the nanosensors are administered. And so you can see quite readily that these breath signals are detectable, and more importantly, they peak rapidly from 10 to 15 minutes after nanosensor administration. So this was extremely exciting to see because this therefore provides a very rapid readout potentially for neutrophil elastase activity. And to show that this activity was specifically generated by neutrophil elastase activity, we then first compared breath signal in healthy mice compared to infected mice. And you can see that the breath signal is elevated due to the increased neutrophil elastase activity. And that's further supported when we introduce a small molecule inhibitor for neutrophil elastase that attenuates this elevated signal. And using confocal microscopy, we were able to confirm colocalization of our nanosensor, which is shown in magenta, with neutrophil elastase in the deep lung. So you can see that here. And you can see the stark contrast between neutrophil elastase activity levels, or I guess neutrophil elastase abundance in the infected lung versus the healthy lung. So having characterized the in vivo activity as well as localized these nanosensors, we then wanted to test these in vivo in a scenario that would more be representative of some clinical context. So breath tests are useful because we can non-invasively collect breath at various time points to potentially monitor disease. And we also know that protease activity levels are dynamic during disease progression. So using our lung infection models, we administered our nanosensors into the lungs of those models at various time points during infection and after antibiotic treatment, and then collected breath and analyzed breath 10 minutes after each nanosensor administration in order to see if we could recapitulate the protease dynamics in vivo. So in our mouse models, we have essentially established infection at t equals 0, and then at day 1, we treat with antibiotics. And this helps us establish a two-phase model where we have acute inflammation within the first day, and then a second phase in which the inflammation resolves over time. And we confirmed that we had elevated neutrophil elastase levels using lung homogenates from a parallel cohort, and that these neutrophil elastase abundance returns to baseline 1 week after infection, after treatment. So when we administered our nanosensors and analyzed breath in the infected cohort, we saw that the breath signal recapitulates the pathology that we observed in the tissue sections as well as the measured protein abundance of neutrophil elastase. And you can see in the healthy control cohort that this breath signal is quite flatline during the entire week. Another potential clinical application for sensing neutrophil elastase activity through breath testing is for alpha-1 antitrypsin deficiency, or AATD. So AAT is an endogenous inhibitor for neutrophil elastase, which means that when a patient has this condition, they're deficient in their ability to regulate neutrophil elastase activity. So what this means in the context of the lungs is that during lung inflammation, there's elevated neutrophil elastase activity that can lead to lung tissue damage. So for these patients that are diagnosed with AATD, they're given weekly prophylactic infusions of purified AAT to inhibit that potential elevations in neutrophil elastase activity. What's interesting is that this type of dosing regimen for this inhibitor has been established based on measured serum levels of the inhibitor after infusion. So once it reaches a threshold level, the patient will receive another dose, which will raise the circulating levels until the next round of dosing. This type of treatment is monitored using functional lung tests as well as imaging methods to monitor for loss of lung density and loss of lung function. And so these types of readouts are quite downstream from the actual damaging protease activity. And so we hypothesized that our nanosensors, which can create this breath readout for pulmonary neutrophil elastase activity, could be used to potentially monitor the inhibition of elevated neutrophil elastase activity, which would therefore be useful to assess new therapeutics for inhibiting this type of activity and potentially new dosing regimen. So to demonstrate this, we turned to genetically engineered models of AATD. And these were a kind gift from Chris Mueller's lab at the University of Massachusetts. So in these mice, we expect that when the lungs are inflamed, that there's elevated neutrophil elastase activity relative to that observed in the inflamed lungs of wild-type mice. And so when we administered our nanosensors for baseline breath reading, we find that wild-type mice and AATD mice, in the absence of inflammation, have the same baseline breath signal. And next, we wanted to challenge these mice with inflammation to look at the resulting breath signals. So in wild-type mice, as expected, when we induce inflammation through an LPS challenge, which is an inflammatory agent, we find that the breath signal is elevated relative to that of healthy controls. And we were really excited to see that when the same inflammatory agent is introduced into the AATD mice, the breath signal is significantly elevated compared to that of the wild-type mice. And so what that means is that we can resolve between this pathology-causing level of protease activity and resolve that from a more normal level of neutrophil elastase activity. And the implications for this is that now we can potentially monitor for the normalization of this activity through AATD or another type of protease inhibitor therapy, which is what we did. So in this fifth group here, we administered a prophylactic dose of alpha-1 antitrypsin and observed that, in fact, the elevated breath signal was attenuated to the same level that we observed in the wild-type mice. So this demonstrates that we can use this type of breath readout to monitor for normalization of aberrant protease activity. So in summary, what I've shown in this initial work is that we can generate rapid breath readouts for pulmonary protease activity using these volatile-releasing nanosensors. And we can use these breath readouts to monitor protease activity that's dynamic during disease progression, and also use these to assess for drug target engagement in protease inhibitor therapies. In the initial work, we focused on developing these nanosensors for a host protease that's secreted during the immune response against a pathogen. But what's interesting is that microbial pathogens also have their own set of proteases that we could potentially develop these nanosensors for. So in subsequent work, we were interested in developing a breath test for pulmonary tuberculosis infection, which is caused by Mycobacterium tuberculosis. So MTB produces this protease called HIP1, which is a virulence factor that allows it to go unnoticed by the host immune response. And so because of the modularity in the chemistries that we use for nanosensor synthesis, we were able to use the same chemistry to modify a HIP1 cleavable peptide substrate with our volatile reporter and demonstrate volatile release in response to the active protease. And to increase disease specificity for this type of microbe, we also wanted to engineer a second nanosensor that was sensitive to an enzyme that is used by the microbe for biomembrane synthesis, and this is known as antigen 85. And this was a collaboration with Laura Kiesling's lab at MIT as well as Deb Hung's lab at the Broad Institute. And so what's nice here is that Laura's lab has previously developed this substrate for antigen 85 that releases a fluorophore in response to antigen 85 activity. So here we were trying to replace that reporter with a volatile readout and translate this into a nanosensor for breath tests for TB. So this together is an example of how we can develop nanosensors for pathogen proteases and enzymes rather than the host side in order to detect or to ID pathogens in the lungs. And another direction that we're interested in pursuing is the application of these nanosensors and breath readouts for non-pulmonary diseases. And this is possible because of the interesting way that VOCs traffic once they're introduced into the body. So they will diffuse from their tissue of origin into systemic circulation, and then after pulmonary gas exchange in the capillary beds, be exhaled in breath. And so what that means is that now we can develop these nanosensors to be delivered through different administration routes, such as through oral delivery or through intravenous or subcutaneous injection, to deliver them to specific tissues of interest. And for us, we're particularly interested in the gastrointestinal space. We think that these probes will be very useful because the GI tract is very abundant with different enzymes, including proteases. And these enzymes come from numerous sources, such as the host, the microbiome, and enteric pathogens. And so if we can develop nanosensors for all of these different enzymes, we can potentially create these disease signatures to monitor gut health. Furthermore, we can use these nanosensors and these resulting breath readouts to potentially investigate how different factors such as diet, age, and drug use affect essential enzymes that are present in the intestine. And then also use these to assess certain pre- and probiotics of interest in our lab. And so currently, we have Vishal, who's a grad student in the lab, working on this with Arsh Patel. And Vince was with us last fall developing C. diff sensors for C. difficile infection in the intestine. So this is just a preview of some of the data that Vishal has gathered sensing microbiome enzyme activities in the intestine. So here he's trying to identify the window of time at which his probes will reach the microbiome in the large intestine and to predict when to expect his breath readout from his probes. So you can see here that enzyme activity from the microbiome is very confined to this large intestine compartment, which was really exciting for us to see. Okay, so with that, I'd like to acknowledge all of our collaborators who have contributed to this work, including Roderick Kuntz and Taswen Ong, who work at MIT Lincoln Laboratories and specialize in chemical detection. They were really instrumental in helping us learn how to detect volatiles and how to process volatile samples. And of course, our collaborators on the TB diagnostics and for the AATD mouse studies. And I'd also like to acknowledge my current lab members, Koi, Vishal, and Arsh, for their hard work this past year, as well as our funding sources. And with that, I'm happy to take any questions.
Any questions?
Yes? Mr. Wagner.
Thank you. In your detection technology right now, you talk about a breathalyzer. Do you actually have a breathalyzer using bench equipment? And maybe you can speak to the technology within the breathalyzer and the detection limits that may be in the handheld versus a lab equipment.
Right. So we have a mass spectrometer in the lab. It's a proton transfer reaction mass spectrometer that we use for these initial studies. So it's, of course, highly sensitive compared to more cost-efficient devices, which the trade-off is lower sensitivity. So we're really working towards sampling approaches that will help us concentrate these volatiles so that we can detect them with more cost-effective devices, such as, for example, a handheld breathalyzer device. So Arsh, who's an undergrad in the lab, is working on those approaches right now.
So I have two questions. The first is, I was curious to know more about the nanocarriers. So it looked like the nanocarriers were being inhaled into the lung, and so I was wondering, did you see any inflammatory response due to this? And like, if so, how do you prevent that inflammation? And my second question is, how do the nanocarriers leave the lungs once there? Like, does it go to any other part of the body? Like, how does that work?
Okay. So the nanocarriers do not trigger an inflammatory response. We know this because of tox studies we've done in our mice. So we've looked at both acute and chronic exposure to the sensor, and have had a pathologist who's blinded to the conditions evaluate the tissues for inflammation, and none was observed. As far as how the nanocarriers cleared, we expect that it's taken up by alveolar macrophages eventually and broken down in those macrophages.
Very nice presentation.
Thank you.
When you introduce the nanocarriers and you do it in subsequent steps, is there kind of a way to optimize the time between each one so you can separate the responses?
Right. That was one of the questions we had going into that longitudinal monitoring study. And based on the prior study where we looked at longevity of the signal, we observed that the dose that we used, the signal was returned to baseline by 3 hours after nanosensor administration. So based on that, we were comfortable dosing as close as in 3-hour increments.
Very nice talk, thank you.
you.
Can you go back to slide 17, please? So I was interested in this in connection with your earlier comment about an LPS challenge in the mice. Is that a lipopolysaccharide?
Yes, that's right.
How is that done?
So that study was in the context of the lungs. So we used intratracheal instillation of a solubilized LPS to deliver that to the lungs to create that inflammation.
And what's the connection of that to the protease?
Right, so LPS is also known as endotoxin. It's from gram-negative bacteria from the membrane, exactly. So we have receptors that are able to recognize those types of agents, inflammatory agents, and that will lead to, inflammation in the lungs.
Yeah, so I know the topic of LPS and like gamma proteobacteria and stuff like that is pretty big amongst the microbiome community with the idea of the loose junctions. And I was curious, whether there could be a connection between the LPS producing bacteria. Well, they're all gram-negative bacteria, as you say, have it, but some I think are more inflammatory than others. And so that's an interesting topic with the whole question of the microbiome, whether you can do any work there, because I think it's very difficult. I mean, the colon, if people do stool samples and look at the genomics and all, it's pretty much the stool sample looks at the colon. But then I think there's a lot of stuff happening in the small bowel as well that there's just no good technique for. And so it's a very interesting set of possibilities, I think, if that could be looked at. So it's very exciting.
I agree. And the small intestine is abundant in many different types of enzymes outside of proteases, such as glycosidases. So that's a subset of enzymes that we're thinking of targeting with the next generation of sensors.
Sounds like a lot of good work possible.
Thank you.
Other questions? All right, I have one. So your released reporter, how do you ensure that itself is not then subsequently metabolized?
Right, so the reporter that we initially used is bio-orthogonal, so we would not expect any sort of metabolism of that molecule in the human body. But that is a fair point for future reporter choice. We would want to identify molecules that are not metabolized in the body. I know of some work where they've used, for example, limonene as a reporter molecule, and that itself is metabolized by the CYP enzymes in the liver. So that would not be, for example, a good reporter molecule to use.
Anything else from the folks here? If not, let's thank Professor Chan one more time.
Thank you.